These notes provide an update to the online video lecture on the central chemoreceptors and are also relevant to the lecture on the chemoreflex control of breathing. I discuss blood flow in the central chemoreceptor environment. This is an important topic because blood flow changes here will affect the CO2 sensed by the central chemoreceptors, and consequently the central chemoreflex control of breathing. This topic is also relevant to the measurement of the central chemoreflex ventilatory response to CO2. Cerebral blood flow is affected by carbon dioxide. This graph shows the increase in blood flow velocity in the middle cerebral artery measured with transcranial Doppler, TCD for short. As the arterial partial pressure of CO2 increases from 25 millimeters of mercury in hypocapnia to 55 millimeters of mercury in hypercapnia, the middle cerebral artery velocity increases in a sigmoidal fashion. You can see by comparing the fitted solid and dotted lines that hypoxia enhances the response to CO2. The sigmoidal shape of the response to CO2 arises from the vascular limitations of vasoconstriction in hypocapnia and vasodilation in hypercapnia. Note, too, that blood pressure, the blue lines in this graph, was also measured and acts as a confounder because changes in blood pressure also affect blood flow directly. In this individual, blood pressure was constant until a threshold PCO2 is reached around 45 millimeters of mercury. Above that, blood pressure and middle cerebral artery blood flow velocity increased linearly with PCO2. Do such increases in blood flow occur in the central chemoreceptor tissue? If so, then CO2 will be washed out as CO2 increases and blunt the response of the central chemoreflex. How can this effect be demonstrated? This effect can be demonstrated using models of the chemoreflex control of breathing, like this one, consisting of 3 compartments. Such 3-compartment models incorporating the washout effect have been proposed for a long time. For example, that of Milhorn, Benton, Ross, and Guyton in 1965, and later Ursino and colleagues in 2001. In these models, it is assumed that the central chemoreceptors are located within a brain compartment whose blood flow increases with CO2. Consequently, as CO2 increases to stimulate the central chemoreceptors, blood flow also increases to wash out the compartment CO2. Thus, the ventilatory response to CO2 mediated by the central chemoreceptors is attenuated. This counterproductive interaction physiology seems counterintuitive. These models must also incorporate the difference between the brain compartment PCO2 and the tissue compartment PCO2. A recent paper by J. Carr and colleagues examined the jugular and arterial CO2 during both Duffin rebreathing test and end tidal forcing tests. As you can see, they showed that the jugular venous PCO2 was almost 10 millimeters higher than the arterial PCO2 during both rebreathing and end tidal forcing experiments. Note that this difference stayed constant during rebreathing but decreased as CO2 increased during end tidal forcing. And this observation is relevant to the measurement of the respiratory chemoreflexes, as we shall see later. To compare the ventilatory response to CO2 with and without an increase in blood flow in the central chemoreceptor region, I made a simple model. Like the one just shown, and models by many others, it consists of the 3 compartments, a lung gas exchange compartment, a brain compartment, and an all other tissues compartment, each with its own material balance equations for CO2. The brain and tissue compartment settings of CO2 productions and blood flows ensured that brain compartment CO2, equivalent to jugular venous CO2, was higher than the tissue compartment CO2, which was equivalent to mixed venous CO2. The upper right graph shows brain compartment blood flow increasing sigmoidally with the arterial partial pressure of CO2, using the cerebral blood flow data from Yamashiro and Kato from 2021. The lower right graph shows the central chemoreceptor control of lung compartment alveolar ventilation, and I used my own equations from papers published in 2000 and 2010. Carotid body chemoreceptors were ignored in this model by assuming a hyperoxic environment that suppressed their response to CO2. The chemoreceptor threshold in the brain compartment was set at 49 millimeters of mercury, higher than the chemoreceptor threshold in the all other tissues compartment of 45 millimeters of mercury to obtain the same resting conditions. Cardiac output increased linearly with CO2 using data from Hewitt and colleagues from 1973. And the upper left graph shows brain compartment CO2 production decreasing linearly with the arterial partial pressure of CO2 using data from Caldwell and colleagues from 2024. The CO2 dissociation curve used in this model used the equation from Milhorn and colleagues from 1965. The model was implemented using the graphical programming language LabVIEW from National Instruments. And here it is in action, comparing ventilation responses to step changes in arterial CO2, that's the dark blue line, from 40 to 50 millimeters of mercury, with central chemoreceptors located in the brain compartment where blood flow, the purple line, increased with arterial CO2, versus responses with the central chemoreceptors in the all other tissues compartment where blood flow increases but little. Note that the brain compartment PCO2, the orange line, is higher than the tissue compartment PCO2, the light blue line. It is apparent that there is a lower and perhaps slower ventilation response, the black line, with central chemoreceptors located in the brain compartment. So indeed, the model demonstrates that if blood flow in the central chemoreceptor environment increases with CO2, the ventilation mediated by the central chemoreceptors is decreased. Does this effect occur in humans? A similar experiment could be done using perhaps indomethacin to decrease the blood flow response to CO2. Indeed, Volsink and colleagues in 1994, shown on the right, found that indomethacin decreased cerebral blood flow, attenuated the cerebrovascular sensitivity to CO2, and stimulated ventilation in newborn piglets. Their experiments confirm the model results and show, at least in piglets, that a decrease in cerebral blood flow in the central chemoreceptor tissue in this experiment induced by indomethacin does increase the central chemoreflex. How is blood flow in the region of the central chemoreceptors controlled? There is some anatomical evidence relevant to the question. As shown here, Hachinsky and Ostergaard proposed that the cerebral circulation should be considered as two different systems. A high-pressure system for the older part of the brain in evolutionary terms that deals with autonomic control. Here, the vascular smooth muscle controlling blood flow must reduce systemic blood pressure to tissue perfusion pressure over a short pathway. Such a blood supply system of high pressure through a high resistance provides a constant flow. By contrast, the cortical low pressure system has a more specialized control of flow distribution, with vascular smooth muscle sensitive to arterial CO2. Indeed, blood flow in the middle cerebral artery supplying the cerebral cortex is higher and more responsive to CO2 than blood flow in the basilar artery supplying the medulla, which has a lower blood flow and is less responsive to CO2. We will see that evidence later. Recall that the central respiratory chemoreceptors are located in the older part, and so the control of blood flow may differ from that in the cortex. However, blood flow control in the medulla remains poorly understood, in contrast to that of the cerebral cortex. Experiments in rats measuring the vascular effects of CO2 in the central chemoreceptor environment provide evidence that blood flow in that region does not increase with CO2. I quote from the paper by Hawkins and colleagues, "Considering that CO2-induced vasodilation would accelerate removal of CO2 and potentially counteract the drive to breathe, we hypothesize that chemosensitive brain regions have adapted a means of preventing vascular CO2 reactivity." They found evidence in rats to support their hypothesis, as shown in this slide. The upper left trace of a rat arteriole in the medullar retrotrapezoid nucleus, where the central chemoreceptors are located, shows a reduction in vessel diameter when CO2 is administered. This result contrasts with the rat cortical arteriole diameter shown in the lower left trace. On the lower right, arteriole diameters from several locations in the brain react to CO2. These rat experiments support the idea that cortical and medullary vascular control differs, and that CO2-induced vasodilation does not occur in the region of the central chemoreceptors in rats. But what about humans? We turn to evidence from human experimentation. Fan and colleagues in 2010 measured both the cortical blood flow response to CO2, transcranial Doppler of the middle cerebral artery, and the ventilatory response sensitivity to CO2 with both rebreathing and steady-state methods of measurement. They used indomethacin to decrease both resting blood flow and the response to CO2. They found that for both methods, the cerebral blood flow reactivity to CO2 decreased with indomethacin. However, the reduction of the cerebral vascular blood flow sensitivity to CO2 with indomethacin was greater using a steady-state method of measurement than using the Duffin rebreathing method. This observation corresponds with the lower cerebral blood flow sensitivity to CO2 when measured with rebreathing compared to that measured using steady-state methods seen by Pandit and colleagues in their 2007 paper, and this difference remains unexplained. While the ventilatory response to CO2 measured with rebreathing was unchanged by indomethacin in Fan and colleagues' experiments, by contrast, end-tidal forcing and steady-state analysis showed that the ventilatory sensitivity to CO2 was increased with indomethacin, a finding also made by Shi and colleagues in 2006, as shown in the upper right. Now, Fan and colleagues argued that with indomethacin, there would therefore be a decreased washout of CO2 from the central chemoreceptor environment when CO2 increased, which would enhance ventilatory CO2 sensitivity, showing that the central chemoreceptor environment was affected by changes in cerebral blood flow. Furthermore, this effect would be apparent using steady-state methods, but not during Duffin rebreathing, where blood flow effects are minimized because of the reduction in the arterial venous gradient of the tissue. These observations therefore suggest that the environment of the central chemoreceptors is indeed affected by the cerebral blood flow response to CO2 when altered by indomethacin. But what actually is the blood flow response to CO2 in the central chemoreceptor environment? Since we have no measurement of medullary blood flow in humans, let's consider the possible blood supply in the region of the central chemoreceptors. As the data from Scow and colleagues show, the posterior cerebral artery, the PCA, and the basilar artery circulation supplying the medulla has a lower baseline flow and a lower sensitivity to CO2 than the middle cerebral artery, the MCA, supplying the cortex. Blood flow in the central chemoreceptor region will likely be less effective, therefore, in washing out CO2. In addition, with a lower resting blood flow, the difference between the central chemoreceptor tissue, PCO2, and the arterial PCO2 will be less. Carr and colleagues demonstrated that CO2 washout does occur for the cerebral cortical circulation, showing that the difference between jugular venous and arterial partial pressure of CO2 decreases with increasing CO2 in steady-state experiments. By contrast, this difference remains constant as CO2 increased during Duffin rebreathing experiments, as we saw earlier. The consequence for measuring the central chemoreflex sensitivity to CO2 is that the sensitivity measured with steady-state experiments should be less than that measured by Duffin rebreathing experiments. Is that so? Here are experiments comparing the ventilatory sensitivities or slopes using both steady-state and rebreathing methods of measurement. Birkenbosch and colleagues in 1989, shown in the upper center graph, undertook the first systematic study of the problem since Reed's original study, which is shown in the upper left. They found that indeed the Reed rebreathing method sensitivities were higher than those obtained using the steady-state method, in contrast to Reed's findings, and suggested that CO2 washout with increasing blood flow was the cause. However, rebreathing studies since then using the Duffin rebreathing method have not found significant differences between steady-state and rebreathing methods of measuring the central chemoreflex sensitivity or slope to CO2. We are therefore left in some doubt as to whether the increase in blood flow with CO2 in the central chemoreceptor environment is sufficient to reduce the veno-arterial PCO2 gradient during steady-state experiments, washing out CO2. So we come to some final words with implications for measurements of the central chemoreflex control of breathing. If blood flow in the central chemoreceptor tissue increases with CO2, the central chemoreceptor ventilatory response to CO2 is reduced due to washout of CO2 as blood flow increases. However, the CO2 response of blood flow in the region of the central chemoreceptors is unknown in humans. In rats, blood flow decreases in response to CO2, in contrast to that of the cortex, where blood flow increases. In humans, the central chemoreceptors lie in medullary tissue, whose blood flow is supplied by the basilar artery, whose blood flow and response to CO2 is less than that of the cortex. Measurements of the central respiratory chemoreflex sensitivity using Duffin rebreathing abolish the washout effect, either by reducing the central to arterial PCO2 gradient or by holding it constant as CO2 increases, so that sensitivity is unaffected. The latter was shown in Carr and colleagues' experiments, which measured a constant gradient with a parallel rise in jugular and arterial partial pressure of CO2 as CO2 increased during Duffin rebreathing. During Carr and colleagues' steady-state experiments, however, The washout effect is evident in cortical tissue, with the gradient between jugular venous CO2 and arterial CO2 decreasing as CO2 increases. Whether the washout effect influences steady-state measurements of central respiratory chemoreflex sensitivity seems to be problematic. Indomethacin experiments decreasing the washout effect show an increased ventilation response to CO2, evidence for the washout effect. But comparing steady-state measurements of ventilation response to CO2 with those obtained by Duffin rebreathing show no difference in slope sensitivity, evidence for a lack of the washout effect. Finally, consider the central respiratory chemoreflex ventilatory recruitment threshold, partial pressure of CO2. When measured with Duffin rebreathing, it is the mixed venous PCO2. However, the actual threshold may be higher by the difference between medullary tissue PCO2 and the mixed venous PCO2. But the resting blood flow in the medulla and medullary metabolism is less than that of the cortex. Both factors will act to decrease the difference between mixed venous CO2 and central chemoreceptor tissue CO2.